kids. So today we're going to do the photosynthesis lab. Alright, so first of all we have to make the chloroplast solution. So what I'm going to do is I'm going to fill a little bit of the blender, cover the blades with just a sucrose solution, nice and icy cold. And then I'm going to take regular old spinach, same stuff that you'd find at the grocery store, and I'm just going to pull that out so I don't blend my hand off. I'm just going to pack that in there. Alright, so now I've got the spinach packed in there. I'm going to blend it up and just 3 10-second bursts. So here we go. So now that I have it all blended, I'm going to pass it through this little cheesecloth. It's got little holes so that the liquidy part can slip through while the chunks stay behind. And so I'll just pour my little smoothie in there. And there it is, nice and green. So here is our solution, basically blended spinach loaded with chloroplast. Delicious. Okay, so now we're ready to actually start the lab. We've got all the solutions we need, and we've got a cuvette here. So the DPIP, remember this is this blue color, and that is going to slowly fade to clear as photosynthesis takes place. I've got water, I've got buffer, and then this is kind of a diluted suspension of the chloroplast solution that I already made. And so now I just follow the lab instructions to set up my cuvettes, and we're ready to go. It's go time. This is the stressful part. So I've got my cuvettes, all 4 of them loaded up, and what I'm going to do is I'm going to get my chloroplast loaded up as well, and then as soon as I add those, this reaction is going to start. So I'm going to start the timer. I'm going to add my drop. Whoops, I think that was 2 drops. Guess we're going 2 drops with this lab of chloroplast, and then I'm going to wipe the side with the Kim wipe, put it in the colorimeter, and I'm going to record the value that it says. So it takes a minute to settle. So 0.407 is my number. So then I'm going to take this out, and I'm going to set it in front of the white light. Now, I'm going to take my next cuvette, and I'm going to wait till my timer hits 1 minute, and then when the timer hits 1 minute, which is 15 seconds from now, I'm going to do the same thing. I'm going to add 2 drops now, since that's what happened with that last one. Almost there. And then I'm going to add my 2 drops. There you go. I'm going to invert. I'm going to wipe the edges. I'm going to put it in there. And I'm going to record that value. And this one is 0.427. It's a little bit different, which is strange, but we'll go with it. And then I'm going to set that one in front of the red light over here. And then I'm going to repeat that process 2 more times. I've got 2 more cuvettes. I'm going to do it again for the blue light, again for the green light, and then we'll check in a minute. Okay, so at this point, I'm going to take my cuvette. It's been exactly 5 minutes now, and so I put that in there, and now I have my second data point. Now, theoretically, it should be a lower number because the DPIP is going from blue to clear. My first number was 0.407. My second number is 0.364. Holy smokes, it's working. So I take that out, I put it back in front of the light, and now at exactly 6 minutes, I'll do the red, at exactly 7 minutes, I'll do the green, exactly 8 minutes, I'll do the blue, and this kind of keeps cycling through every 5 minutes until I get all my data. Okay, so as you can see, we turned off the light shortly after taking that last little video clip so that it was only the right color light that was exposed, that the cuvette was exposed to. And then we collect our data. So we're going to post that data online so you can access it. And then note that you're going to make what is called a linear regression. In other words, you're going to graph the data, and then linear regression is basically the slope. And then the slope is a measure of how fast the reaction took place. So if you see a really shallow slope, that means it took a long time for the reaction to happen. If it's really steep, then it happened really quickly. And notice that since the color is fading, it's going from blue to clear, absorbance actually goes down. And so all of your slopes are going to be negative. And so the most negative is the fastest rate. And so basically we're going to take the absolute value of all those slopes so that the most negative number, the absolute value of that would be the highest number, which is the highest rate, which is how it should be. So basically you're going to graph the data, you're going to calculate the slopes of each of your lines, and then you're going to report that as an absolute value so that you can easily compare the rates of reaction. Good luck.